LTP is a long-lasting enhancement in signal transmission between two neurons after repeated stimulation. Reveals various structures that are involved in LTP. On the postsynaptic cell are both Nmda and Ampa receptors. These receptors are usually localized together at many postsynaptic sites and are activated after the binding of the neurotransmitter glutamate. The Ampa receptor is permeable to sodium ions. The Nmda receptor is also permeable to sodium, but more importantly, it has a high permeability for calcium. The Nmda receptor is also blocked by magnesium, which prevents the ions from passing through the receptor. When an action potential travels down the Schaefer collaterals, it leads to the release of glutamate, which binds to both the Nmda and Ampa receptors. When a low-frequency action potential is propagated down the Schaefer collaterals, a small amount of glutamate is released. The Ampa receptors will open and allow an influx of sodium into the CA1 postsynaptic cell. This causes a slight depolarization event in the postsynaptic cell. The glutamate also binds to the Nmda receptors, but no ions will pass through the pore due to the magnesium blockade. While the small amount of neurotransmitter release signals a response, it is not enough to cause LTP. This is comparable to you studying material for only a few times. You will not activate a high-frequency action potential from studying for just a short while. When a high-frequency action potential travels down the Schaefer collaterals to the terminal, a larger amount of glutamate is released from the presynaptic terminals. This is comparable to you studying for longer periods. This results in higher frequency action potentials. When glutamate binds to the Ampa receptor, a greater depolarization event occurs. The Ampa receptors remain open longer due to the increased concentration of glutamate, which allows a larger amount of sodium to enter through the Ampa receptors. This influx of sodium causes a large depolarization event in the postsynaptic cells, which repels the magnesium blockade from the Nmda receptors through a process known as electrostatic repulsion. At this point, the Nmda receptor, with glutamate bound, allows sodium and calcium to enter through its pore. In this way, Nmda receptors are known as coincidence detectors because they require a presynaptic and postsynaptic event for channel opening, the binding of presynaptically released glutamate, and a significant postsynaptic depolarization via activation of the Ampa receptors. As previously, early phase and late phase. During the early phase, the calcium binds to its respective binding proteins and causes the insertion of new Ampa receptors onto the postsynaptic cell membrane at the active CA3-CA1 synapse. These influx of calcium causes an increase in transcription factors, ultimately resulting in gene expression and new proteins to be synthesized. Some proteins resulting from this process include Ampa receptors, which are inserted into the postsynaptic cell membrane at the synapse. In addition, there is an increase in the synthesis of proteins called growth factors. These are involved in the formation of new synapses, which is the basis for synaptic plasticity. Your brain can change as you learn. These synapses are formed between the CA3-CA1